Thank you. So to change topics completely from what Paul just presented, I'll be showing work we've carried out over the course of this project the past 5 years. Environmental impact of diluted bitumen, impact environmental du bitume dilué. And I showed various highlights over the past few years, and I'll change it up again this year. So just some background again. So in 2019, crude bitumen production from Alberta's oil sands regions totaled around 3 million barrels per day, compared to 0.5 million barrels a day for conventional crude. So yeah, moving bitumen is very important for Canada's economy. And to transport bitumen via pipeline, it is blended with lighter hydrocarbons, yielding a less viscous diluted bitumen. Now, there's a lot of different types of dilbit terminology, but here I'll just simplify it and call it dilbit. So although pipelines are generally considered safer than other means of transport, major ruptures have occurred. For example, the famous one that took place in the Kalamazoo River in 2010. So dilbit spills may behave differently than conventional crude spills, yet our understanding of the environmental impact of dilbit in freshwater systems, particularly groundwater, is limited. There's been a lot of work done by various groups for the past 5 years that have actually helped to address some of these research gaps, although the work done in groundwater systems is still pretty sparse. So I'll show you all the activities that are within this project. The main goal, of course, being to better understand the relationships between geochemistry, hydrology, microbiology, and toxicology during natural attenuation of dilbit using controlled spill experiments in the lab and field. So this covers a wide variety of different disciplines. And there's people from I show the main players on the first slide, but there's a lot of people involved in this project. And so there's two main components. One, looking at shallow groundwater systems from controlled spill experiments that took place in large columns, 2 large tanks, and 3 glass tanks in collaboration with people at CanMet. Nicholas Udding. And 2, a smaller component, but still very interesting and very important, looking at dilbit in river and lacustrine systems under 1 wave tank wave action weathering done with collaborators at CanMet, and 2, microbial uptake of spilled dilbit in the lake sediments. And that was carried out in collaboration with people at the Experimental Lakes area in Northwest Ontario. So today's talk, I'll just be showing some highlights carried out over the work carried out over the past year in the large column experiments. So you've probably seen these slides before. So we had large columns that were approximately 1 meter or 1 meter length and 60 centimeters diameter. And we carried out these controlled spill experiments in Labo Lourdes. We had 2 columns, which we amended with dilbit, 2 with conventional heavy crude, and 1 as a control. And they were carried out under controlled temperature conditions at around 10 degrees Celsius over several months. And so we collected the leachate in the bottom. We also watered these, of course, to represent the average precipitation that from the area where the soil was collected near Mont-Saint-Hilaire, near Montreal. And we also adjusted the pH to reflect that of the slightly acidic rainwater. And the 9 degrees I mentioned before was to reflect spring and autumn recharge. And so we analyzed a wide variety of parameters in the leachate that was collected in glass bottles. You can see that were connected with sealed with Teflon tape. And so total organic carbon, total inorganic carbon, BTEX, benzene, toluene, ethylbenzene, xylenes, PHs, polycyclic aromatic hydrocarbons. So those 2, the PHs and BTEX, are often considered the main toxic constituents from oil spills. And so we also analyzed acid extractable organics, which includes naphthenic acids. They're a major component of bitumen. They're the most common contaminant in oil sands tailings ponds. Of course, the geochemistry of tailings ponds is very different, and why you have naphthenic acids at high concentrations there is different than what you find in the spill. But nonetheless, we decided that might be important to analyze. Major elements and metals, and of course, we did toxicity analysis on fathead minnow. So there's also the soil core component, which you've probably seen this presented in various presentations over the past few days. And so in this, we analyzed, we collected cores, 10 cores over the course of the experiment. And the holes were filled in with bentonite to maintain the hydraulic integrity of the system. And we analyzed TPHs in the sediments and the soil, phospholipid fatty acids, PLFAs, which are markers for the active microbial population. We carried out compound-specific carbon-13 analysis and C14 analysis to understand the microbial uptake of petroleum hydrocarbons. And we carried out microbial community analysis and pH concentrations. And what's currently ongoing, it's a bit due to some delays from COVID, et cetera, and instrument problems. We still haven't quite finished the compound-specific carbon-13 and deuterium analysis of pHs. But this work was recently published in a paper in Environmental Pollution. This was part of the MSc thesis of Leah Mindor, who's now actually a full-time employee in the Delta Lab. It's great to have her. And so the main conclusions of this study were that dilbit and conventional heavy crude showed similar biodegradation over the 104 days of experiments, and up to around 20% of carbon in microbial PLFAs were derived from either conventional heavy crude or dilbit. That's the graph that's shown on the right there. The C14 values decrease over time in the PLFAs, showing a greater incorporation of petroleum carbon, which has a value by default of minus 1,000 per mil. The top line at the top, the dashed line at the top corresponds to the carbon-14 value of the soil that we use in the columns. So in terms of the microbiology, the abundances of polar monas, a known hydrocarbon degrader, increased over time. And so this result, the main result was that natural attenuation potential for dilbit was similar to conventional crude following a bad ozone spill. So I'm not going to talk any more about that. I'll just show you some highlights from the leachate experiments. So I presented some of these slides a couple of years ago, but now we have some results to add to this, which are very interesting. And so this is the plots of the day post spill on the x-axis versus concentration of total organic carbon and TIC from the control and the average of the two dilbit columns or two conventional heavy crude columns. And so you can see over time, that you see an increase in the TOC corresponding to the petroleum hydrocarbon constituents that are reaching out into the water and nothing in the control, a lot lower relative to those two columns. And you have TIC, which is reflective of the biodegradation and microbial uptake of petroleum hydrocarbons you saw in the previous slide that I showed the highlight for the column work. Nothing surprising there so far. So as I mentioned, BTEX and polycyclic aromatic compounds, PACs, also incorporates pHs, are generally considered some of the two most toxic constituents from most petroleum hydrocarbon spills. And so you can see here the BTEX, which are monoaromatic hydrocarbons, more water soluble than heavier molecular weight PAHs or PACs. They kind of spike out in the cold lake blend bitumen, dilbit, and the conventional heavy crude columns relatively quick after a couple of weeks. And due to sorption and mainly due to sorption in the soils and within the oil itself, pHs, which were very, very low, by the way, kind of like start to leach out over time. And note from the much higher concentration of BTEX, of course, which was quite high and above guidelines after a couple, when they spiked after a couple of weeks. So here's the toxicity results. So on the left is the proportion of malformed larvae for fathead minnows in the control, dilbit, and conventional heavy crude. The asterisk indicates a result that's statistically significant above the control value. So you can see on the first 3 time sampling periods, the water was quite toxic, and similar levels of toxicity in both the cold lake blend dilbit and the conventional heavy crude. And on the right axis, we have the relative CYP1. So CYP1 gene is known to be involved in phase 1 detoxification of petroleum hydrocarbons, and it's well established as a highly sensitive biomarker for oil exposure, specifically for dilbit and conventional crude. So similar to toxicity, you see this increase in this expression of this gene in both the Cold Lake blend dilbit and conventional heavy crude higher than the control. And it's interesting to note that the highest toxicity or gene expression for a known gene for metabolizing petroleum hydrocarbons was seen at around in day 34. And so, as you saw from the previous slide, VTEX and pHs weren't responsible for that. So what was responsible for this increase in toxicity? So here's the trace metals on the left over time versus the AEOs, so the acid extracted organics containing naphthenic acids on the right. And so you don't see any increase or any change at all in the trace metals compared to the control. So you can negate that, dismiss that as a potential source of toxicity. And on the AEOs, you don't really see much increase relative to the control until the very end of the experiment. And these are still very, very low concentrations, but there is obviously a difference. You have some components, acid extractable components, that are increasing from the leachate over time in both the petroleum and both the conventional crude and dilbit columns. So taking those analyses, which are carried out by Orbitrap mass spec at John Headley's lab in Environment Canada, and looking at it further. So this is breaking down these are the main class species within the acid extracted organics fraction over time, and plotted with the main species in the control, the Cold Lake blend dilbit, and conventional heavy crude. And so you can see they have very similar profiles in the main species. The O2 species you can see there in the middle corresponds to classic naphthenic acids. And they don't show any substantial differences between the actual control or the petroleum compounds, petroleum columns. But you can see here, if you look at the oxygenated sulfur species, the sulfonic acids, the O2S and O3S species, you can see that they increase over time at similar levels in both the dilbit and conventional crude. You don't see a spike at day 34. So these compounds, which are increasing in the leachate from both petroleum columns, is likely not responsible for the toxicity effects that we saw. So that's still a mystery, unsolved, but we can actually, we have provided more insight into which compounds potentially don't cause toxicity, or and also how the spill, how its geochemistry evolves over time in the Vado zone. And that's it. So I'd like to thank you for being here for the presentation, and any questions I'll be happy to answer.